Good morning, good evening, or good afternoon, depending on where you're joining us in the world today. Welcome to the first of 2022, the first Nano Explorations. My name is Brian Anthony. I'm the Associate Director of MIT.nano, and I'll be your host. Today, it's my great pleasure to introduce Soumya Pratap Tripathy. We'll kick us off with talking about maybe something that you've heard a little bit about over the last year or two, this bizarre little virus called COVID-19, this esoteric thing in the world that Soumya's decided to do some sensing on. Now, just kidding. Yeah, Soumya's done some very interesting work in using waveguide sensors for the sensitive detection of COVID-19. Soumya is a graduate student at MIT in the media lab. And Soumya, I'll turn it over to you. Participants, this is being recorded. Please feel free to chat your questions in the Q&A. I will try to answer some questions in real time. If clarifications are needed, I'll interrupt Soumya as necessary. And then we'll have a Q&A more rigorously at the end after Soumya is done. But feel free to put your questions up as we go. Soumya, I'll turn it over to you. Thank you for your time today.
Thank you, Brian, for the introduction. So today I'm going to present on my master's work, that is peptide beacon integrated planar-waveguide-based sensor for low pulse, rapid, and highly sensitive detection of COVID-19 viruses. Before starting my presentation, I want to thank you, people like Professor Joseph and Jacobson, for the guidance during my master's work. Thank you, Brian, for suggesting me to present on this Nano Exploration platform. I also want to thank Professor Ed Boyd and Professor Joseph Paradiso for the guidance to my master thesis. And I want to thank you, Dr. Pranam Chatterjee and Manmita, for helping me in this project. I also want to thank you, MIT.nano facility, MTL, CBS, BCS, and MRL, CMSC, and Koch Institute, where I have used all the facilities to make this work on. So everybody know COVID-19 pandemic started in 2019. Since then, we have different kind of tests in the market. If you want to categorize those tests, you can have two categories. One is nucleic acid-based test, and another one is antigen antibody-based test. So in case of nucleic acid test, the limit of detection is near 1 attomolar, that is 1 copies of ml, and the sensitivity is more than 90%. And the turnaround time is 1 to 2 hours, and it needs expertise to handle. However, in case of antigen or antibody-based test, the limit of detection is near femtomolar, within 1 to 100 femtomolar, and is less sensitive than RT-PCR with a turnaround time less than 30 minutes, and is easy to handle, and you can do it at your home. So hence, there is always a trade-off between limit of detection, sensitivity, and turnaround time. So the more sensitivity of nucleic acid test is due to the identification of target before testing it. And in case of antigen and antibody test, do not amplify before testing. So there is a room to develop a diagnostic platform that would be low cost with tens of dollars, and it will give rapid turnaround time at less than 10 minutes. And should have a single step detection of the COVID-19 virus without complex biochemical amplification reaction. And it should have sensitivity, specificity, and limit of detection comparable to RT-PCR, which is the gold standard for all kind of biomolecular test. So to deal to it, rapid and rapidness, and to divide of complex biochemical reactions, we have developed peptide beacon-based test, where we have developed one peptide that has quencher and fluorophore two of its end, and the two ends of the peptide make the coil field stem that make this plate happen between quencher and fluorophore. So in the place of loop, we have target binding peptide. So once that loop binds with the target, it makes conformational changes in the whole peptide and separate the fluorophore from the quencher in space. So once the fluorophore go away from the quencher, the fluorescence output of the system increases. So by using this increasing fluorescence after binding a target to the peptide beacon, we have developed this peptide beacon-based diagnostic platform. So there are already literatures based on peptide beacons, and there are sequences that make cross a coil in the literature. So we took different sequences from the literature and put our spike binding peptide in between, and saw the structure in Tia rosetta and Rosetta ab initio. And both of them, in Tia rosetta and Rosetta ab initio, gave us the structures in the closed conformation that predicts that the quencher and fluorophore will be near to each other. Then we docked this peptide beacon to the spike protein binding reciprocating domain and saw that. So once that S-RBD binds to the peptide beacon, the two ends will go away from each other and make the on state. So after this computational selection, we wanted to see whether our peptide beacon candidates are actually binding to the S-RBD or not. So for that, we performed one test that is based on one chip delta TPR E3 ubiquitin ligase molecule. So we fuse our peptide beacons with this chip delta TPR E3 ubiquitin ligase and subjected to degradation of S-RBD GFP. So this chip delta TPR E3 ubiquitin ligase actually degrade this S-RBD GFP that shows that the molecule that is fused with this molecule are like binding to the S-RBD. So it took one positive control that is nanobody NB20, and the A2N peptide is the actual peptide that binds to the S-RBD, and that's in our loop region. So this result suggests that among three peptide beacon candidates, the C17, A2N, C21 induces the most level of degradation of S-RBD as GFP, and followed by C21, LC22, and C13, LC21. So this motivated us to test this peptide beacon in vitro. So to synthesize these peptide beacons, we took two sets of peptides. One peptide contains the quencher, that is dibesyl, and another sets of peptides contain the loop region that binds to the S-RBD and the fluorophore. So after coupling to these two sets of peptides, we purified our actual peptide beacon candidates through sequential Malditov mass spectroscopy, SPLC, and further Malditov mass spectroscopy from the purified material from the HPLC. So after getting purified peptide beacons, we try to see their response in the presence of S-RBD. So you can see among the three peptide beacon candidates, the C13LC21 has negligible response toward S-RBD and influenza hematogglutinin. And the C17LC21 has the pronounced response toward S-RBD with a limit of detection near 20 femtomolar, and followed by the C21L C21 with a limit of detection of 400 femtomolar. After this, to deal with the cost and high sensitiveness of our device, we adapted the technology of torch microscope. So the torch microscope is the total internal reflection fluorescence microscope used for high resolution and single molecule imaging. So in torch microscope, we use total interreflection to create confined evanescent wave. So the confined evanescent wave excitation within 50 to 200 nanometer of the surface create substantial reduction of the out of focus signals and like background noise. That is the reason TORP is used for single molecule imaging and high resolution imaging. So here we miniaturize that torch microscope to make our Nano waveguide-based device, and we integrate with the single photon sensitive silicon photomultiplier as a for the sensitive fluorescence readout. So in this device, we coupled one collimator laser beam to the waveguide with a coupling angle of 38 degrees that makes the collimator laser beam to incident at the polymer cladding and core interface at 66 to 0.27 degree. That is more than the critical angle of the core cladding pair. So that and it makes the TIR to happen. So due to that, TIR will get the luminescent wave on the glass core. So the tough device has two parts. One is cartridge that contains the planar waveguide, laser, and the CPAM, and another one is the readout module. And the readout module contains the CPAM amplifier circuit and other ADC circuits to for the signal acquisition. And the cartridge cost around like $30, and the readout module is about $100. So after making this cartridge, we want to see whether there is luminescent wave on our device or not. So for that, we took different fluorescent beads. We immobilized those beads on our waveguide and imaged that under the confocal microscope. So it took a G-stack imaging. So in case of luminescent wave-based excitation, as the luminescent wave is confined above about 50 to 200 nanometer of the surface, the microbeads only on the surface will give fluorescence, not other microbeads in the solution. But in case of epilumination, all the microbeads that is within the path of the laser will give the fluorescence. So here you can see in this video on the left, that is a minute of illumination. You can see there is like only fluorescence on the surface microbeads, not in the volume. But in case of epilumination, there is fluorescence on the surface beads and other new microbeads in the volume. So this determines that our minute top has luminescent wave on the surface. So after confirming there is luminescent wave on our monitor, we went to see how our device is performing on the basis of like detecting fluorescence signal from the surface bound molecules. So it took biotin content coupled to FITC and try to see at what concentration our monitor can detect the fluorescent molecules. So our monitor can detect the surface bound fluorescent molecule at limit of detection of 100 attomolar range. And this motivated us to couple peptide beacon on the surface of our mini-top and try to diagnose the COVID-19 from the samples. So we immobilized ceftaroline on our planar waveguide, and we immobilized the peptide beacon through a biotin calculator. And once the virus come near the peptide beacon, so it opens up and the fluorescence output from the peptide beacon increases. So then we synthesize another set of peptide beacons that contains the biotin with original sequences. That is C17 LC21 and C21 LC21. Then we try to purify that peptide beacons using some HPLC and Maledov mass spectroscopy. And further, we try to detect the COVID-19 viruses on our monitor integrated with peptide beacons. So at first, we try to detect the spike protein receptor-binding domain on our monitor. So you can see the C17L, C21 peptide beacons can detect the S-RBD at a limit of detection of near 500 attomolar, and the other peptide beacons, C21L, C21 can detect the peptide beacon also in the range of 500 attomolar. So RT-PCR has a limit of detection near 1 attomolar, but our device is now performing at the region of 500 attomolar. There is like a lot of work need to be done to get to the point where RT-PCR is performing. So also we try to detect the pseudoviruses on our monitor using both peptide beacons integrated on our device. So our device can detect the pseudovirus particles at about like 10,000 copies per ml of range. And however, the RT-PCR can detect like 1 copies of virus in 1 ml. Still a lot of work need to be done to optimize this work. So in summary, our device can perform like one-step detection of SARS-CoV-2 spike proteins and virus particle with sub-femtomolar limit of detection. And the turnaround time of our device are in the region of 10 minutes, and it is rapid. And we have also established a pipeline for developing peptide beacons for other viruses. And the Minitab platform can be used for on-site handling for the samples. And we are on the path to do the clinical test of our Minitab and to scale up the production and go for the startup. So this is all on the topic that we can integrate with planar device for the local rapid and sensitive detection of the COVID-19 virus. Thank you.
Soumya, thank you. So my virtual and physical applause here. Participants, please feel free to put your questions in the chat. I was going to kick it off with this question that just came in. How does this, Soumya, compare to other low-cost biosensors for COVID-19 and or other viruses? Maybe if you could put it in context in terms of where you see the use scenarios, the viable use scenarios for the approach, and also, yeah, putting it into context of other approaches.
So if you see, so there is two kind of approaches. One is molecular approach, and another one is the device-based approach. Or you can combine both molecular approach and with device-based approach. So the key reason is that in case of RT-PCR, if you even have one copies of the viral mRNA in 1 ml, and you put all the enzymes and all the recombination molecule in that 3D volume, and that amplify the target, and you at the last you detect the fluorescent output. But in case of antigen antibody test, we do not amplify, and we just do the test in rapid manner, and that is the reason we do not have more sensitive output as RT-PCR. So if you were to see this peptide beacon technology, can be applied for other viruses as well, and we have the pipeline for that. So we have immobilized this peptide beacon on our device surface that make in difficulties in having that facilitate that does not facilitate that detection in the 3D volume as we do in case of RT-PCR. And there is need to be done some kind of mass transport optimization to bring those target to near the surface so that our peptide beacon can detect them. So if you took this molecular approach, so I'm planning to develop some kind of a split enzyme technology that will detect the peptide, the target, and amplify the signal, not through the RT-PCR, but some kind of chemical reaction that will give us more output in the fluorescent or in the observance. I think so.
Good. Soumya, thank you. So another question here. Can you please elaborate on the peptide beacon S-RBD binding domain and how binding causes a change in the conformation?
Yeah, so in the peptide beacon, can I go to the slide?
Please do. Yeah.
So the peptide beacon has two parts. One is the stem part and the loop part. So in the stem, there is one coil peptide. So this peptide make the coil structure so that the quencher and fluorophore will be in the coastal distance where the freight can happen. So in the loop is the binding peptide that bind to the target. So once the target bind to the loop, that creates the conformational strain in the whole peptide structure that makes the coil structure to break. And once the coil structure is broken, the fluorophore and quencher go away from each other, and that makes this fluorophore separated from the quencher more than its posterior distance, and the freight efficiency decreases, and make the fluorescence output increased from the system. So this dwelling between low fluorescence state to high fluorescence state upon binding the target to the peptide beacon is the core technology in our case.
Very good, thank you. Another question from Mohammed here. Probably a question on everybody's mind. Is this approach, is the sensor sensitive to different variants? And what if the virus goes through additional mutations? What do you expect there are the changes that would, how sensitive and specific can you be?
Yes. The peptide beacon we have developed here is like can detect the whole family of SARS viruses, like including the previous SARS viruses and like SARS-CoV-2. So also we have like one library of different kind of like peptides that specifically bind to the different variants of COVID-19 viruses. If you like incorporate those peptide in the loop region, this peptide beacon can be developed specific to different variants. So we are in a way to do that, and like yeah, it will take time. Thank you.
So maybe this is a question related to maybe sort of how this fits into a larger system. Brian asks, what challenges would be faced in applying this technique to biological fluids, which include some complex proteins and other chemical background?
Yes, we have not tested this peptide beacons with that complex biological fluids. So obviously that will need some kind of like optimization in this like peptide sequencing and structure. So we are like approaching that problem using like deep learning and like machine learning based design of these peptide beacons, where it will be only specific to the target, not to the other proteins. So that is like one concern, like we are working on that.
So a question, I think maybe this is a clarification question, Soumya. Mark asks, your presentation, it appears as though you're using microbeads. Are you using microbeads? And if so, how large are they? And what is the interaction in the very thin evanescent zone? So I might want to just clarify how you were doing, what the microbeads are doing for you.
Oh, so we just like use those microbead to taste whether there are evanescent waves on the surface or not. So these microbeads are like about 1 micrometer in diameter. So this microbeads generally give fluorescence one excited by the laser beam. So in case of evanescent wave, since the evanescent wave is only confined to the surface, the microbead on the surface will give fluorescence, not other microbeads in the volume. So this is a method to confirm whether there is evanescent wave or not. So this microbead does not like bind with peptide beacon, or like we do not like take this microbead as our target. This just to like confirm the presence of evanescent wave on our device.
So the microbeads were a calibration characterization technique to verify function of the device. Very good, thank you. Morgan asks, as a follow-up question, are there state-of-the-art low-cost sensors that are similar to your approach, and how does what you're doing differ?
Yes, so let's say like there are different kind of like antigen antibody test where people use some kind of like protein or like some like aptamer like molecule that bind to the target. Let's say if you want to take the like any kind of like antigen antibody test, so there are based on like gold nanoparticles coupled with antibodies and within a matrix where you put out your sample, and the target will go and bind to the antibodies and give you like color output. So here also we are using that kind of like technology, like the peptide beacon that bind to the target, but that like keep the fluorescence output not in like changing color. So that fluorescence output can be detected using single photon sensitive detectors such as like CPAM. And that makes our device more sensitive than those antigen antibody tests. So if you take those antigen antibody tests that can be like used at the home and like in large scale. So also like our device in the same manner can be used in large scale at home, I think.
Very good, thank you. Let's see. So maybe this is a related question. I think you touched on a little bit. If the first question of part A is, did you do any clinical tests? And the second part is, what approaches do you envision to obtain rapid turnaround time that are maybe more system related? But yeah, sort of, what's the state of clinical tests, and how do you get to the rapid turnaround?
Yes, like as I told, like this work is a part of my master thesis, so did not got much time to do the clinical test, but we are in the collaboration with some companies, those provide clinical specimen to test with. So we are in the plan to make this device in like some large scale and send these devices to them so that they can test with the clinical sample. And like to get to that rapid thing and with limited detection here to artificial. So we really need to take some approaches where we need to do some like mass transport optimization so that the target come more rapidly to the surface to be detected by the peptide beacon.
Thank you.
Thank
you. So there's one sort of detailed question here, then I'll close it off with a sort of a high level question as well. Brian asks, what is the binding energy of the coiled stem and what is the binding energy of the target solute?
Yeah, there's like very like good question. Like, so actually the peptide beacon design, this optimization has been like done by the Manvita and like Pranam. So they have the data with those binding energy. So I was not like put all this data here. So I can like make like I can connect you with those guys and you can get all the data from them, I think.
Thank you, Soumya. And I guess maybe if you could sort of comment, reflect a little bit on, I think it may have been, for some may be missed that you're using a technique called of integrated photonics. So this waveguide. I was wondering if you could comment a little bit about the sort of the manufacturing issues associated with, there's sort of the instrument issues, but what are the issues that you need to be aware of or that could influence performance or long time shelf life as they relate to how you actually would fabricate these and
yeah, so here all the planar waveguide was like fabricated by hand, like it took like glass slides and like dice them using dicer and like coated all the polymer cladding by the spin coating and immobilized structure on the surface. So it's a big chip. So if you in the like millimeter scale, like 1 centimeter by 2 centimeter. So if you really go to the chip scale on like let's say we fabricate all the like burst waveguides or like waveguides on the silicon wafer and immobilize all these molecules in a like integrated manner, I think we can like increase the like we can like make this technology scalable. And like also like instead of this like planar waveguide technology, if you can use some kind of like resonator based approach such as like ring resonator or like WGM and other technology, we can like really increase the sensitivity to like atomic levels.
Very good. So I think with that, I think we've covered, there were a few questions, but I think they're redundant. I didn't check them off, but I think, so Soumya, I want to again send you the virtual and the physical applause and participants. Thank you for the great questions and the dialogue and your participation. Soumya's talk and all prior talks over the last couple of years can be found on the Nano Explorations YouTube channel. You can hit the MIT.nano webpage to get linked to that. But I want to share with you the next talk coming up in just under a month on Tuesday, February 8th. We'll hear from Devin Beck from Lincoln Laboratories, MIT Lincoln Laboratories, on 3D printing of glass with metastable silicates. So that's coming up as the next Nano Exploration in February. Again, Soumya, thank you, everybody. Thank you for your time and attention. Stay warm if you're in the greater Boston area. It's very chilly today, but stay safe, everybody, and be well. So thank you.
Thank you.